All living things are made of the most basic units of life, cells. In order to sustain all life on Earth, organisms need to be continuously making new cells. This is done through reproduction, a process where something makes more of its own kind. This is a quick lesson on cellular reproduction. Pictures were illustrated by Tanya and narration is by Milin. Before we begin, there are a few vocabulary terms we should go over so that we have a clearer understanding of the processes ahead of us. Let's start with chromosome-related things. A haploid is a cell with one set of chromosomes. A diploid is a cell with two sets of chromosomes. Since a chromosome is a gene-carrying combination of genetic material and protein being tightly held together, homologous chromosomes are simply a pair of similar chromosomes, one maternal and one paternal. Sister chromatids are two identical copies of a chromosome. This can also be called a duplicated chromosome, and the point that the sister chromatids are connected is called the centromere. A daughter chromosome is a sister chromatid or unduplicated chromosome that has been separated from its pair during cell division. Lastly, centrioles are organelles that create mitotic spindles to aid cell division and are present in animals only. In the cell, there is an organelle called the nucleus that stores the genetic material, DNA, and regulates cell functions by directing protein synthesis. The small darker spot within the nucleus is called the nucleolus, which makes another type of genetic material, RNA, which assists in protein synthesis as well. The genetic material inside the nucleus specifically is called chromatin and is a condensed form of DNA. This is later condensed further into the chromosomes we were just talking about during cell division. Lastly, we have centrioles, which produce mitotic spindles, and a pair of them is called a centrosome. There are two ways that human cells reproduce, asexually and sexually. Asexual reproduction is the production of offspring from one parent cell into two identical daughter cells. Sexual reproduction is the production of offspring from the fertilization of an egg by a sperm. Some key differences between mitosis and meiosis are that mitosis happens in body cells, is asexual reproduction, contains only one cell division, and results in two identical diploid daughter cells. In meiosis, however, sex cells are sexually reproduced through two successive divisions, resulting in 4 unique haploids. This is an overview of the eukaryotic cell cycle. It's split into 2 big phases, as you can see in the outer rim of the circle, the interphase and mitotic phase. Interphase, where the cell grows and prepares for division, is split into 3 subphases. First is G1, or first gap, where the cell increases its size. Next is the S, or synthesis phase, where DNA is synthesized or replicated and the number of chromosomes double, each with a pair of sister chromatids. Last is the G2, or second gap phase, where the cell is given time to grow again. The M, or mitotic phase, is where the cell divides. In mitosis or meiosis, the cell's organelles and structures align and prepare themselves to divide, and in cytokinesis, the cytoplasm is split and the cells are completely separated. Now we'll talk about the processes of mitosis and meiosis. Meiosis is uniquely composed of 2 parts, meiosis 1 and meiosis 2, so we'll go over meiosis 1 first. Both begin with parent cells, which are just cells that will divide into daughter cells that can be different or identical. The first step in meiosis 1 is prophase 1. In prophase, 2 homologous chromosomes, which are these cyan and blue X-shaped things here, overlap and cross over their chromatids, exchanging DNA. The site where the chromosomes cross over is called the chiasma, and the structure formed by the 2 homologous chromosomes is called a tetrad. Prometaphase is actually a short phase between prophase and metaphase that's often looked over. At this point, the nuclear membrane breaks down to release the chromosomes, and spindle fibers attach to them on their kinetochores, which is just a spot they connect on the centromere. The next step is metaphase 1, where the tetrads will organize themselves randomly along the metaphase plate, which is an invisible plane that the cell plans to split at. This random combination of tetrads is called independent assortment and creates diversity in the daughter cells. Next is anaphase 1, where the tetrads are divided back into homologous chromosomes and pulled apart by spindle fibers to opposite sides. Remember that these spindle fibers are made from centrioles and connect to the kinetochores on the centromeres of the chromosomes. Telophase is the last step of meiosis 1. This is where the nuclear envelope and cleavage furrow is formed, which is this pinch between the dividing cells. Usually at the same time, cytokinesis occurs and we end meiosis 1 with 2 different haploid daughter cells. We will now explain mitosis and meiosis 2 simultaneously side by side, since they're essentially the same process, but meiosis 2 begins with 2 haploid cells, while mitosis starts with 1 diploid. We begin the process with prophase in mitosis or prophase 2 in meiosis 2. In this step, the chromosomes condense and centriole pairs or centrosomes create mitotic spindles. Just like we talked about in meiosis 1, prometaphase occurs between prophase and metaphase. The nuclear envelope dissolves and spindles attach to kinetochores on the chromosomes. In metaphase, the homologous chromosomes align themselves along the metaphase plate once again. Notice how there's no crossing over or independent assortment in mitosis or meiosis too. This explains why mitosis produces no genetic diversity. The next step is anaphase, where the chromosomes will split into two sister chromatids and are pulled apart by retracting spindles to opposite ends of the cell. The cell also elongates and prepares to split. Lastly, telophase occurs where the cell further elongates and the cell membrane forms a cleavage furrow. Again, the nuclear membrane forms back around the chromosomes and the spindles disappear. Cytokinesis is not really a part of meiosis or mitosis, but it does occur as the last stage of the M phase of the cell cycle, following either mitosis or meiosis. Going into more depth, a ring of microfilament made of actin and myosin will contract and pinch the cleavage furrow until the daughter cells separate. We should also mention that for plants, cytokinesis is completely different. The plant forms a cell plate in the middle of the cell, which is constructed from Golgi vesicles. Going back to animal cells, both mitosis and meiosis began with one diploid parent cell, but mitosis ends with two identical diploid daughter cells, and meiosis results in 4 different haploid daughter cells. As you can probably deduce from the numbers, mitosis contains 1 division, while meiosis contains 2 successive divisions. Now let's do a quick recap on the advantages and disadvantages of the 2 types of cellular reproduction. Mitosis, a process within the asexual reproduction of a cell, is fast, efficient, and independent. However, it lacks genetic variation, which makes it difficult for cells to adapt and evolve to changing environments and conditions. Meiosis, sexual cellular reproduction, produces diverse offspring through crossing over and independent assortment, but it's reliant on a cellmate, has few daughter cells, and is much slower than mitosis. Before we finish, we'll talk about an example of artificial gene editing. A cool thing that scientists are experimenting with is recombinant DNA. They combine parts of DNA from different species into a single DNA molecule. This method is commonly used in genetic engineering. Now it's finally over. I hope you've enjoyed and learned from this lecture. Here are our references for information, and please like, comment, and subscribe.